Hello friends, let's discuss the Renin-Angiotensin system today. The Renin-Angiotensin system is a very important physiological mechanism for the maintenance of blood pressure and the electrolyte composition. And it also has a very important pathophysiological role in the etiology of hypertension. So it becomes a very important system which is targeted by means of various antihypertensive drugs, and I will just outline the relevant physiology and some pharmacology of the drugs which are used to control this renin-angiotensin system. So starting with the participants in this system, the first one is there is this hormone or this renin. This renin is actually a substance which is secreted from the kidneys. That's why it is called as renin, because the origin is from the renal. The origin is renal. That's why it is called as renin. And this renin, what it does is it causes cleavage of one substance called as angiotensinogen to angiotensin 1. This reaction is catalyzed by renin. Okay, so this angiotensinogen, it is a plasma protein secreted by the liver, and it keeps on circulating in the plasma unless it is acted upon by renin for its conversion into angiotensin 1. And what happens is the amino terminal, 10 amino acids, they are cleaved from angiotensinogen to form a decapeptide called as angiotensin 1. Okay, so now I will start with the rest of the story, and that is the angiotensin 1. It is a decapeptide, and the sequence of this, or I can just write angiotensin 1. Now angiotensin 1, its sequence is aspartate, arginine, valine, tyrosine, isoleucine, histidine, proline, phenylalanine, histidine, and leucine. This is the sequence of angiotensin 1. This angiotensin is also called as angiotensin 1 to 10. This is another name for this angiotensin. Now this angiotensin 1 is acted upon by means of one enzyme called as ACE, or you can call it as angiotensin converting enzyme. So this ACE, what it does is it cleaves these two, the last two amino acids from the sequence, and forms what we call as angiotensin 2. Okay, so if we call this as 1 to 10, this is the amino terminus. You can number it from here: 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So now the two amino acids from the carboxy terminal, they are removed by means of this ACE, and leads to the formation of angiotensin 2. So since 9th and 10th are gone, now angiotensin 2 can also be called as, or you can call it as angiotensin 1 to 8. Okay, so this is what normally happens. And this angiotensin 2, then it can have various fates. One thing is, if acted upon by an aminopeptidase. Aminopeptidase means something which cleaves the amino terminal. So you can get another substance called as angiotensin 3, or you can call it, since you have now removed the first one, it would also be called as angiotensin 2 to 8. Okay, and this angiotensin 3 may be further cleaved by means of an aminopeptidase. I can name that as AP. So aminopeptidase again cleaves this into another substance called as angiotensin 4, and this angiotensin 4, since again now the second is gone, so it can also be called as angiotensin 3 to 8. Okay, and this angiotensin 1, not only by means of ACE but by means of another enzyme called as ACE2, this can be converted into another angiotensin which cleaves only one from the carboxyl end. So that would be called as angiotensin 1 to 9 by means of a single a carboxypeptidase, you can call it. And this angiotensin 1 to 9 can again be acted upon by ACE, forming, now ACE will remove 2, so it will become angiotensin 1 to 7. So now we have got, now this angiotensin 1 to 7 can also be bought by, it can also be formed by directing this angiotensin 2 by means of a carboxypeptidase to this side. Okay, so now what we are having here is various angiotensins. One is angiotensin 1, another is angiotensin 2, then angiotensin 3, angiotensin 4, and angiotensin 1:7. Okay, so there are 4-5 types of angiotensins, and these angiotensins, they are important because they have important roles in the pathophysiology of hypertension. Now the most important angiotensin in all of these is angiotensin 2. This is the most important one. Why? Because it acts upon one type of receptors, what we call as receptors, which we call as, it acts on a receptor called as AT1 receptor. Okay, it also acts on another set of receptors, what we call as AT2. But the more important one is the AT1 receptor, and the roles of AT1 and AT2 receptors are more or less opposite to each other. But the important, pathophysiologically, the most important receptor is the AT1 receptor, which is actually responsible for the hypertensive effects of angiotensin 2. Okay, angiotensin 3 also acts on both these receptors, but with a little lesser propensity. Okay, the angiotensin 4, it acts on its own set of receptor. It is called as AT4 receptor, and it is specific for angiotensin 4. And this angiotensin 4, it has a different role from the Renin-Angiotensin system. Its role is in cognition and memory. So this type of angiotensin is more formed in the brain. Then angiotensin 1 to 7, this angiotensin also has a role which is opposite to that of angiotensin 2. Okay, so this role is also different from the angiotensin 2. It has a totally separate role, and it has its own set of receptor called as a mass receptor. So now we have got some idea about the angiotensin 1, angiotensin 2. Angiotensin 1 is not very effective. It has to be converted into angiotensin 2 before becoming effective. So angiotensin 2 is the most important agent which is formed in this process, and which is responsible for the hypertensive effects. Okay. Now what is the function of this system? The function is to regulate the blood pressure and to regulate the electrolyte balance. Now how does it do it? One important thing is angiotensin 2 by itself is a very potent vasoconstrictor. It causes vasoconstriction of the vessels inside the body. So when it causes vasoconstriction, obviously it increases the blood pressure. Okay, and another thing what it does is it leads to the stimulation of a very important hormone called as aldosterone. Now, aldosterone is released by means of uh, the stimulus provided by angiotensin 2. So that also makes it very important. Now aldosterone, the role of aldosterone is to reabsorb sodium and to release potassium into the urine. So reabsorption of sodium, again, whenever there is reabsorption of sodium, there is also reabsorption of water. So that leads to increase in the blood volume. So all these mechanisms, what they do is they tend to increase the blood volume and the blood pressure ultimately. So those all these effects. Then angiotensin 2 again has one more effect, that is it increases the sympathetic outflow. So it increases the sympathetic tone inside the body. So when the sympathetic tone is more, again vasoconstriction occurs. So all these things, they ultimately lead to an increase in the blood volume and in the blood pressure. So this is a normal physiological function, but whenever this function goes beyond what is needed, it becomes pathological. So that leads to problems such as hypertension. Okay, now how is this system stimulated? What is the stimulus for release of this system, or you can say the stimulation of the system? We need a stimulus, and the stimulus is a decreased tubular load to the tubules. The tubules, they get a decreased sodium load. Whenever the sodium is less in the tubules, it is sensed as a state of a decreased blood volume or a decreased blood pressure. So the body tries to normalize it, and when it tries to normalize, what it does is it causes release of renin from the juxtaglomerular cells. We call them as the JG cells, which are present near the glomeruli. And what tells these JG cells that you have to release renin is the macula densa, which senses the concentration of sodium which has been presented to it. And this is in the thick ascending loop of Henle. When it crosses just close to the glomeruli, that region is specialized to sense that, and that region is called as the macula densa. So macula densa sends signal to the JG cells accordingly what is happening inside the body. For example, if the sodium load is less, it will stimulate the JG cells to secrete renin, and if the sodium load is more, it will suppress that. So it is like a balance between the two. Whenever the sodium load is less, you will have more of renin. Whenever the sodium load is more, the renin-angiotensin system will be suppressed. Okay, so one very central thing is the release of renin from the JG cells. Okay, so now coming to the drugs which affect this system. So there are 3 types of drugs. For explaining the drugs, I will again make the system so that it becomes easy. So what do we have? We have angiotensinogen. Angiotensinogen, it is converted into angiotensin 1, which is again converted into angiotensin 2, which now acts on the AT1 receptor. So this is a simple chronology, you can say. So angiotensinogen is converted into angiotensin 1 by means of renin. Okay, angiotensin 1 is converted into angiotensin 2 by means of ACE. Okay, so this is how the things go down, and then AT1 receptor is stimulated, and this receptor leads to increased blood pressure, increased aldosterone, increased sympathetic activity, and also feedback, and it is also responsible for the negative feedback. So negative feedback, which suppresses, this negative feedback will go back and it will suppress the renin release. So this system is like self-sustaining. Whenever the angiotensin 2 levels are up, they will suppress the renin release. So it is a negative loop. Okay, so now how do the drugs affect here? There are the first drugs which were synthesized to control this were the ACE inhibitors. So these are the ACE inhibitors, or you can write them as ACE-I or ACE inhibitors. They block this step. When they block this step, the synthesis of angiotensin 2 is suppressed by means of ACE. Okay, now what happens then? This Renin-Angiotensin system is stopped at the level of angiotensin 1. Since angiotensin 2 is not produced, AT1 receptors are not stimulated, so all these effects, that is increased blood pressure, increased aldosterone, increased sympathetic tone, they are all suppressed. Okay. But the problem with ACE inhibitors is that there are also some mechanisms what we call as the non-ACE pathways. These non-ACE pathways, by means of that, the angiotensin 1 can still be converted into angiotensin 2. Okay, so even though we have given ACE inhibitors, the person may still have a higher BP. So the hypertensive control may not be that good. So in order to counter that, another type of drugs were synthesized, and those drugs were called as the angiotensin receptor blockers. Means they are the blockers of this. They block the angiotensin receptors, AT1 receptors. So they are antagonists at the AT1 receptor. So even though angiotensin 2 is produced, it is not able to act on the AT1 receptors because you have given an antagonist. So those drugs, also we can call them as ARBs. These ARBs, they provide you a more complete blockade of the system. Why? Because they inhibit this receptor. Now it does not depend, whatever may be the source of angiotensin 2, it will not be able to act on the AT1 receptors, and it will not cause all these problems which can eventually happen in a hypertensive. Okay, and there is another group of drugs which acts here at the level of renin, and this group of drug is called as a direct renin inhibitors, and these direct renin inhibitors, they inhibit renin. Now they block the conversion of angiotensinogen to angiotensin 1, so they block it at the very at the very first step. So all these three types of drugs are there which are used for suppressing the renin-angiotensin system. Now important thing about ACE inhibitors is that the ACE enzyme also leads to the formation, also lead to the breakdown of bradykinin. This ACE enzyme is also responsible for breakdown of bradykinin. Now bradykinin is a small molecule, small peptides which are responsible for, whenever this bradykinin is not broken down, whenever we are giving ACE inhibitors, the bradykinin levels, they rise, and they lead to some side effects with these drugs. Important side effects are, one is or dry cough, very commonly seen with ACE inhibitors, dry cough, and another is angioedema. So these are the side effects which can be seen with ACE inhibitors. But there is also one beneficial effect, that is bradykinin by itself has a vasodilatory action. So owing to that vasodilatory action, uh, the antihypertensive effect of these ACE inhibitors is potentiated. Okay. Now that benefit is not provided by the ARBs because they do not interfere with the breakdown of bradykinin. But ARBs they block this AT1 receptor here, and whenever this AT1 receptor is blocked, this angiotensin now it binds to the other set of receptors, that is the AT2 receptors, with more propensity, because the AT1 receptors they are occupied. So this AT2 receptor, it has got actions which are more or less opposite. So it has got antihypertensive effects, it has got anti-proliferative effects, so all those beneficial effects. So that's why whenever you are giving ARBs, you are expecting more stimulation of AT2 receptors, and this leads to beneficial effects. So it is always debatable whether to use the ACE inhibitors or the ARBs or the DRIs or the direct renin inhibitors. Mostly the tussle is between ACE inhibitors and the ARBs. So the ideal thing is to start with ACE inhibitors unless the patient complains of dry cough or angioedema, and if the patient is unable to take the drug owing to these side effects, you can switch over to the ARB. I hope that is enough. I will continue with more videos on this topic later on. Thank you so much.